Hello, I'm Professor Paul Bingham. This is Biochemistry 1. Our topic in this segment is gluconeogenesis. So this is yet another of the last few pieces we need to see the entire picture of human metabolism, and is the very last piece we need to see the complete picture of glucose metabolism. So let me remind you of gluconeogenesis in context. So this is all the events that either feed into or feed away from glucose 6-phosphate, the activated form of glucose that enters glycolysis and the pentose phosphate pathway, for example. Here shown here in the center of this diagram. Gluconeogenesis is in some sense the opposite of glycolysis, with some interesting differences that we'll focus a lot on in the next few minutes, allowing you to start with pyruvate, or as you'll see in a few moments, the equivalent of pyruvate, and rebuild glucose out of it. So why might you do such a thing? You burn glucose to pyruvate to get energy. Why are you going to turn around and spend energy to turn pyruvate back into glucose? Well, the fundamental answer has to do with this need for homeostasis that we've talked about in the last several segments. So many tissues, brain and muscle in particular, need a constant supply of glucose to function properly because of the unique properties of glucose as a catabolic intermediate that we've talked about before, its speed and its oxygen independence, for example, in the case of muscle. Many tissues are absolutely dependent on a continuous supply of glucose. And so sometimes if the diet is heavy in protein and lipids, for example, then the body, in particular the liver, has to rebuild from the breakdown products, for example, amino acid breakdown products of protein, has to rebuild glucose out of that to keep the blood glucose levels constant. So the liver sits there monitoring the blood all the time, releasing glucose when it needs glucose, and absorbing it and storing it when it doesn't. So gluconeogenesis is the fundamental way in which amino acids, for example, are incorporated are converted back into this absolutely vital asset of glucose. So let's put that in context a little bit more. So you can think of gluconeogenesis roughly, as we've said, as converting pyruvate, rebuilding glucose out of pyruvate, converting pyruvate back into glucose, obviously for energetic, thermodynamic, fundamental reasons. You have to input energy into that process. Pyruvate has a lower free energy, 2 pyruvates do than the 1 glucose you'll build out of them. In fact, let's look at where the pyruvate comes from. So one place it will come from is lactate. Now, the reason we care a lot about that is that you recall when we talked about muscle metabolism earlier, and we'll return to this in much more detail later, when we're exercising very vigorously, our muscles go into oxygen debt. That is, they don't have enough oxygen to feed our energy needs. And so, in fact, we run glycolysis through the pyruvate, dump the extra reducing potential that we can't absorb and transfer to oxygen as we normally would onto lactate. The lactate is released from the muscle into the circulatory system and then reabsorbed by the liver, which together with the kidney is the only organ that can do gluconeogenesis, and then rebuild that back to glucose, release it back into the circulatory system, where, among other things, go back to the muscle again. So it becomes a process whereby the muscle can expend energy, and then the liver can take the resulting lactate and rebuild it into glucose to resupply the muscle. One important source. Another important source, as we've already talked about, is amino acids, either from dietary sources, if we're eating a particular high-protein diet, or if we're starving or fasting. The body will release fatty acids from lipids, of course, but eventually those will be used up. And in fact, muscle protein will be hydrolyzed, released as amino acids back into the circulatory system, taken up by the liver, and turned back into glucose. One last thing is the capacity of the liver to take its TCA cycle, its citric acid cycle intermediates, and run those back into glucose. That's not done through pyruvate, as we'll see in the next few minutes. And that's, in fact, done in a slightly different way. But as you'll see, it's all part of the same process. It all fits together. I've already alluded to it. Let's make it absolutely clear here that gluconeogenesis is the exclusive purview of the liver and the kidney. And, in fact, the liver is quantitatively much more important. It goes on in the kidney, but it's the liver that's the big the big business actor here, the guy who has enormous capacity to do gluconeogenesis and should be thought of as the central player that is absorbing things like amino acids from the circulatory system, turning them into glucose and releasing them again. Very closely related to the liver's ability to also supply ketone bodies, as we talked about before. The liver should be coming into focus as a central organ in the management of energy metabolism, and in particular the metabolites in the circulatory system that feed everybody else. Okay? All right. And again, the liver is especially important in the process of in the course of doing this. As we said earlier, the liver is also the only organ that does glycogen synthesis on the really large scale that makes it possible to feed other tissues, not just store energy for its own future use as other tissues like muscle and brain do. So again, the liver is really central here to understanding glucose metabolism, and again is unique in its ability to do gluconeogenesis, the thing that we're going to talk about in the next few minutes. Okay, so let's look at an overview of the biochemistry of gluconeogenesis, and then we'll come back and look at the reaction mechanisms that are involved. So again, these are the things that can feed the citric acid cycle intermediates, lactate or amino acids can all feed pyruvate into the process, or the equivalent, as you'll see. In fact, oxaloacetate turns out to be a crucial intermediate in gluconeogenesis. At first glance, you might be scratching your head and saying, wait a minute, this is gluconeogenesis. As you might expect, it's going on in the cytosol of the cell. What is oxaloacetate, a mitochondrial citric acid cycle intermediate, doing in all of this? Where did that come from? Out of the blue, it sounds. As you'll see, it actually has a very specific role to play in this process. And so this is a slightly more detailed picture of gluconeogenesis. Pyruvate from either lactate or amino acids is converted into oxaloacetate, or oxaloacetate is exported from the mitochondria into the cytosol from the citric acid cycle. In either case, oxaloacetate is a crucial intermediate in rebuilding glucose from pyruvate. More about that in the next couple of minutes. There is the structure to remind you of oxaloacetate. So let's now see where oxaloacetate is coming into this, what's going on. This is a complicated diagram, and as usual, we will zero in on specific parts of it as we need to. In green on the right-hand side is glycolysis running top to bottom. And on pink on the left-hand side is gluconeogenesis running bottom to top, the opposite of one another. And what you'll notice is that there are a number of shared reactions. I'll call your attention to those in a moment. And then there are reactions that are unique to gluconeogenesis, distinguishing it from glycolysis. So let's look at that a little more carefully. Notice what we're doing. We are, roughly speaking, starting with pyruvate and rebuilding glucose out of it. And notice here that on the gluconeogenic side, oxaloacetate is part of this pathway, unlike glycolysis, in which oxaloacetate has nothing to do with it. The reasons for that will become a little more obvious in a moment. So, in fact, there are steps that distinguish gluconeogenesis from glycolysis. Each of them is boxed here. So, going bottom to top, the first is pyruvate carboxylase, as we'll see. Then there is a pep carboxykinase, and then nearer the top there are two phosphatases. Don't worry about absorbing those details right now. I'm just trying to, we'll look at each of them in detail over the next few minutes. What I'm trying to call your attention to here is that there are lots of steps in common that we're not boxing, and then there are a few steps that are unique to gluconeogenesis that distinguish it from glycolysis. And as we'll see, what those steps are designed to do is to pump energy back in so that we make pyruvate to glucose itself a net exergonic process. Okay? All right. Notice where the energy inputs come here. So two of the energy inputs, by the larger boxes in red in the middle, are in fact just the reverse of glycolytic steps, where instead of reducing a NAD coenzyme, we oxidize it. Or instead of making an ATP from ADP, we make an ADP from ATP. That is, we're just running those reactions in reverse. But the other two at the bottom, notice the bottom center boxed in red, are the two unique steps that pump energy into gluconeogenesis, making that upward reversal of glycolysis actually itself exergonic and therefore possible to run efficiently. Okay, so let's look at the first of those two steps at the bottom that are designed to make gluconeogenesis exergonic, energetically doable, favorable, drivable. Okay. So again, here is this.